Hi, I'm Kat Navarro, Lee with the B Foundation for Brain Aneurysm Prevention, and I'm here with Dr. Andrew Hale, who's a resident at the University of Alabama, Birmingham.
Thanks for having me.
Every year, the B Foundation does a grant cycle for preventative research, and you were one of the grant recipients last year, and that's how I met you at the gala. I know a lot
Yeah.
of these grants are funded by families whose lives have been affected by a brain aneurysm, and this grant was funded by the Yezi family. Before we really get into that, I'd love to just talk more about you so that we can get to know you better. And I'd love to know how you got into neuroscience and neurosurgery.
Yeah, so thank you so much for having me. I really appreciate the B Foundation and their support. I grew up in Hudson, Ohio, which is in kind of Northeast Ohio area. And really from about the middle of high school, I knew that I wanted to become a doctor. And so I ended up going to Case Western Reserve University in Cleveland, Ohio, for my undergraduate work and majored in biochemistry there. I joined a lab pretty much as soon as I stepped on campus, kind of with the idea of just, getting a little bit of lab experience because I knew that that was something that would be helpful for medical school applications. But it turned out to be something that I really developed a passion for. And so my first laboratory experience, I was in a lab that was in the cardiology department, and we were studying how blood vessels form, and we were trying to understand the cell signaling and transcriptional mechanisms that regulate how different blood vessels kind of form and take their identity. And so that was kind of my first foray into research. And then because of that outstanding experience, I knew I wanted to go to medical school, but I also wanted research to be a real significant and meaningful part of my career. So I ended up applying to combined MD and PhD programs. And so I ended up going to Vanderbilt University in Nashville, Tennessee. There I earned my PhD in biochemistry. Once COVID hit, there wasn't, much going on, and the hospitals were, it was hard to kind of get rotations in towards the end. So I ended up doing a brief postdoctoral fellowship in human genetics, and really was trying to think about, how do I use my background as a scientist? How do I utilize all of this kind of emerging and evolving technology to understand something about, neurosurgical patients? I was really fortunate at Vanderbilt to have some awesome neurosurgical mentors there. And then I ended up matching into neurosurgery at the University of Alabama at Birmingham. And I'm interested in pediatric neurosurgery, but also in cerebrovascular neurosurgery. And so we started thinking about ways to, combine my background in genetics with some, unique patient samples that we can get from patients with cerebrovascular disease. And that kind of led us to this project. And so really the main crux of what we're trying to study and understand is the hypothesis is that there's something fundamentally different about the way the cells that comprise an aneurysm are made up of compared to the rest of the cells in your body. So whether that be, the immediate vessel segment that precedes or is, that is distal to the aneurysm, or something that is inherited. So whether or not that is a DNA mutation or something that's in your genome that is different between, patients who develop aneurysms and people who don't develop aneurysms. And then moreover, we know that, aneurysms tend to run in families. And so the rationale that there is a significant genetic component to those families and their predisposition to develop that disease is very high. So, that's kind of a long way to say, I really had a outstanding kind of first experience in cardiovascular biology, which then kind of took me full circle to now trying to use some of that same information, but combined with my kind of molecular and genetic side that I learned while I was at Vanderbilt to really, think about how an individual patient's aneurysm or cerebrovascular pathology may be different. And if we can identify anything within those or any genetic abnormalities within those lesions to potentially lead to new treatments.
Yes, that is, research that so many people would really, find helpful because I know a lot of times when you meet somebody with a brain aneurysm, the first thing that people say is, how did you know? Or a lot of times there's so much mystery around it where people don't
Yeah.
understand that, first of all, that they even have one. Most of all, a lot of times people don't know that they have one. And then it just there seems to be a lot of mystery around it, except for those cases where people get checked because they know that they have family members who have it.
Exactly. And so there's been a lot of studies on kind of the genetic basis of aneurysms, but they've almost entirely relied on analyzing, blood or cells from the inside of your cheek to look at kind of inherited factors in what's called your germline to see whether or not those genomic factors or those DNA variants or mutations predispose you to risk of aneurysms or are a primary cause of aneurysm formation, or do they predispose you to have, rupture and subarachnoid hemorrhage? I kind of liken this to a car, right? There's a million different parts that go into a car, and each individual component plays a very specific role. And you can take certain pieces out and the car might run okay, but then you take that one key piece out of that engine and the whole thing just gets derailed. And so
Mm.
genetics is a little bit like that, where you're trying to find that critical missing piece that really then changes the biology in a significant way.
Thank you so much for explaining it in that way, because I know that sometimes with, neuroscience and explaining the research, people who aren't familiar with that can get overwhelmed or might not understand. So having that analogy is very helpful.
Yeah.
What would make this project successful for you?
I think really the main thing that we're interested in now is that we've reached kind of a critical point in cerebrovascular biology where we pretty much know the pathways that are involved. I mean, in general, we have a pretty good understanding of the developmental, transcriptional, cell signaling pathways that control the way a blood vessel or an endothelial cell or a smooth muscle cell, we kind of know the critical components of what allows that biological process to occur in a normal way. And so what I think now is the challenge is translating those discoveries into meaningful data for an individual patient. And so what do I mean by that? So, each person's aneurysm is going to be different. Any cerebrovascular lesion, anyone's healthcare is a unique experience and their disease is unique to them. So what can we do on the neurosurgical side and on the scientific side to capitalize on, knowing something very specific about the biology of an individual person's aneurysm? Okay. So one way to do that is this emerging kind of technique that has been really exciting. It's called endoluminal biopsy, where a foil that is deployed into an aneurysm or into a vessel segment is deployed as it would be normally. It's rested against the endothelial cell layer, which is the cells that are in direct contact with blood as it flows through the vessel. And you get some of those endothelial cells that inevitably stick to that coil. And so when you take that coil out of the body, we can do a number of different things in the lab to isolate those cells and then ultimately learn something about the genetics of that individual cell. Okay. And so really, I think the main goal for this project, and where I think a lot of the field is starting to really think critically about and how to translate that information that we can get from genetic sampling of a sample like that and start to think about how do we use that information to actually change treatment for someone.
What would the next steps be like with, after the research? Is this something that is then rolled out to different hospitals, or how does that work on your end?
I think there's a few different kind of critical steps that need to be taken in order for any of this technology to be able to be used on a wide scale basis. Like I said, we kind of know the genes that are really important, but developing a, clinically scalable, diagnostic platform that can identify those key mutations is something that I haven't seen that's available yet. And so starting to think about how do we kind of create this platform so that we can start taking the information that we gain at in the lab and actually use it to help an individual patient that we see in the hospital.
Well, just knowing that, that it's being worked on and technologies like this and research like this is constantly developing is, very hopeful for a lot of people. We live in an
Mhm.
age where we're lucky to have these in the works.
Yeah.
Is there anything else you want to share about what you're currently working on?
No, I mean, I think that's the
Yeah,
relevant part that's to the funded work. But I think that there's a lot of other exciting things I think going on in the field of brain AVMs and similar kind of technology and approaches for utilizing genetic information to improve care and maybe even tailored drug therapy is something that is frankly, right on the horizon. I think it's something that we could very much see utilized in the next few years. And so I'm really kind of excited about that.
me too. Well, thank you so much, Andy. We really appreciate this. And I think a lot of people are going to be very hopeful about the research and the project that you're working on.
Well, I appreciate the invitation. As always, it's been great to connect and chat with you again. And thank you very much to the B Foundation for kind of setting this up and keeping us together.
Thank you so much.